Okay, guys, thanks for coming. Thank you for those who came to my talk the other day. There were two lovely people. I don't know, there were only like 5 in the whole room, so I give you props for showing up. 6 o'clock's hard. My stomach was growling. Okay. Last lecture before the exam. This is going to be probably short and sweet. I was talking to my collaborator who I'm submitting a grant with, and I was like, God, my lectures suck so much right now because I have no time to work on them because I'm busy writing a grant. And he's like, just explain that to them. And I'm like, something tells me that that's not going to be a good excuse. But so I'm explaining that to you. But the grant goes in Monday, which means things should vastly improve because I won't be living colon 5 all the time. Okay. This is about global chromosome structure. A lot of this you've learned before probably, and we'll learn it a little bit more deeply. What? Is there a discussion that we need to talk about? There was just a bunch of murmuring. No? Okay. Okay, business. Exams next week, if you didn't know that. Sorry. It's not going to be like last week where I said that and then I changed it. That's like a once-in-a-lifetime experience. I've actually never changed an exam in my life. I've only been teaching for a few years, so I guess there's always the future. There is a quiz this weekend. I really don't want to give you one because I don't have time to make it. But I need to give you one because we need to have enough quizzes so you can drop some. So if you want to not have one and lose an opportunity to drop when you did badly on, you can put in your comments right now, and I'm happy to not give you a quiz. But I think that you guys would be happy to have like a semi-simple quiz and drop a poor grade from earlier. So we're going to go with that. Okay, the other thing I want to talk about is trying to stay on topic. And I've talked about this before in class, but there have been a lot of comments from other students that people are feeling a little frustrated that the conversation keeps going right here and no one can hear what's going on, and it's usually off topic. So those of you guys who have a huge passion for science, have a lot of great things to talk about, come to my office hours. We can talk for hours about chicken DNA, or whatever we want to talk about, we can do it. But for the rest of the class, we are going to try to stay on topic as much as we can. And my apologies to those who've been feeling frustrated about that. Please always come to me with your comments and whatnot. Okay, and I only say this section because this is really where you guys are all sitting. Okay. So far, so this is I was trying to come up with my list of things to focus on for the exam. And this is pretty much what we've covered. We did experimental methods. Make sure you understand them. And especially the assays that we did papers on in recitation, they are beyond fair game for a short answer essay type question. So really make sure you understood that. Go back over your notes. You spent 2 hours talking about it with other people, so you should know them. Yes?
So with those, what kind of, you ask these questions, if you are trying to figure out this, what kind of experiment would you use?
No, that's not very challenging at all. No, I would ask you something like, you're doing this assay, you get this data, what does it mean? So if you understand the assay, you should have no problem interpreting the data, and you've already done it before with my help. So it's fair warning. Make sure you get it or come see me. Any questions about that? Does anyone want to cry? Yes. Can I post the past quizzes again? I can open them up for the next few days, yes.
Can you? Huh? You can see them on Blackboard. If you go to your grades and you click on your score, it shows you the whole quiz and answers.
Oh, okay. So I don't have to. Thank you so much, because for every semester, people always ask me to open them, and they open them. Then I have people who are trying to retake the quizzes, and I have to deal with that. Okay, so apparently on Blackboard, if you go to your grade and you click on it, you can see your quiz. Let me know if you have trouble. So assays studied and talked about in recitation and lecture, so make sure you understand that. And they're directly related to what we've learned about in class, so it kind of makes sense that I might want to ask you more about them. Chromatin structure and function, modifications on histone tails, modifications on DNA, what enzymes are involved, effects on gene expression, complementation assays in yeast to make sure you understand that, all aspects of it, and centromeres, telomeres. We spent a day on each of those, and then today we're going to talk about the larger scale organization of chromosomes. Okay? Everyone feeling good? You know exactly what you have to study, so you all get an A. Global chromosome structure. Yeah, there's no one here today, am I right? Did everyone go to Hopkins? Is that what's going on? There was a trip to Hopkins. Oh, stupid lights. Okay. You're gonna hear me say that every single day. Instead of doing something about it. Chromosomes are folded into large loops of chromatin, and I was thinking this is where that off-topic discussion came up the other day, possibly. So these are called lamp brush chromosomes, probably because they look like lamp brushes. Who's ever brushed a lamp before? I'm thinking like they must be talking about like when you dust your lampshade, maybe like a Swiffer. That's what they have nowadays. So they're called lamp brush chromosomes because they have these loops. So I think this is more than one probably. I was trying to look. There's probably one. But they've got these loops of DNA looping out here. This specifically is in amphibian oocytes that are developing. You can only visualize them under the microscope when they're these chromosomes from amphibian oocytes, not from our cells. They're much smaller, but it's a model for studying. So it's used to study interphase structure of chromosomes. And I want to point out interphase structure, not mitotic structure, which is where we looked at the telomere staining. Those were mitotic chromosomes. So my question for you guys, or whoever I'm going to pick randomly, what is the purpose of all of these loops? If you just had to think about these loops. So the way to think about this is that in developing oocytes, you're going to have lots of gene expression, right? And this is where you see this more than anything. Pierre. There you go. So what do you think the purpose is of all of these loops? Okay. Do you want to phone a friend? No, you don't want to phone a friend? Okay. How about Alexis? Yes.
Would it allow for greater area in terms of the base pairing, so it can allow for greater
Transcription. I heard a magical word that I was looking for. Transcription. Thank you for your bravery. Yes. Probably has something to do with more access to DNA that's going to be actively transcribed. Does anyone want to add to that? Yes?
Would it have anything to do with modifications, like more area modifications to improve proteins?
More area for modifications. You mean histone modifications? Well, you might think it might be controlled in a way by histone modification. Yes? The loops keep your surface area great, but your overall size is small. And does that have to do with transcription? Yep. Yep.
Would it kind of lead to package it in a way?
Right, exactly like what we just heard up here. It has to do with packaging, so you can keep your surface. Yeah, so your surface area is great. So you have access to more areas for transcription, but you're still keeping it slightly condensed so you can fit in the nucleus. Right? Yep.
So what about replication? Since it's at the beginning of meiosis, right? So DNA has to be replicated to start meiosis. So do you think replication of DNA?
Well, so this is interphase. So you have S phase during interphase. So you're right.
Oh,
Right. So this would change slightly for DNA replication, right? Because you would need to open everything up
little by little.
little by little. Yes. Yes. Not all at once. Right. Any other thoughts? Okay. Good. Okay, so this is what the structure of the lamp brush chromosome looks like. It is, so it's believed to represent that of all eukaryotic interphase chromosomes. So even though we're talking about amphibians, it's just a model for studying interphase chromosomes. So you have your lamp brush chromosomes, and this is actually two homologous chromosomes close together. And then if you look at this one region here of the single chromosome, you get that looping lamp brush kind of look. And then they're held along this axis at the chromomere. And then if you look at the individual loops, you get these extended loops of chromatin. So, as someone said just a few minutes ago, histone modification and the effects on subsequent effects on chromatin structure, decondensed area. My typo, decondensed area is required for gene expression, right? So you've got this looping, and then you've got your histone modifying enzymes, chromatin remodeling complexes, RNA polymerase, all those things we've learned about. Those are going to come in. There's greater access in this loop, right? They're going to come in and modify and even further decondense that chromatin and allow for gene expression. Any questions or thoughts? Okay, so you can actually, yep.
So you said that the loops were for gene expression?
Well, they're further decondensed for gene expression. So the looping allows for access to regions of the chromosome, and then you can modify and further decondense the DNA, allowing for gene expression. So we're going to look at the histone modifications here. So here they were using Drosophila polytene chromosomes, and they were studying different histone modifications. And so in A, here, this is histone H3 lysine, oh, I forgot to put the K in. Lysine 9, sorry, it's not H39. Lysine 9, trimethylated, is that right? Yep, trimethylated, and it is stained green. So all of that is a methylate, it's a histone, H3, with H3 tail is methylated on lysine 9, okay? And then the DNA is all red. And then here, it's same lysine is acetylated, and you can see it's in a very different place, and the DNA is red. So this is all acetylated histones over here. And this right here is pericentric heterochromatin. So it's highly, highly condensed heterochromatin. Would that make sense that it's associated with methylated lysine on histone tails? Yep, why not? And if you notice that you don't see any of the green here, this is all DNA being stained. So there's no acetylated lysine modified histones on in the heterochromatin. Does that make sense? I want to see some. Yes, good. Three people nodding. Does anyone want to nod this way? Okay, because you guys all have had lysine modifications, histone modifications kind of drilled into you. So this is kind of just proof of principle. Very cool, right? Yes. Can I repeat it? Sure. Thank you. Thank you for speaking up. Yes, so the green here is methylated histones, and the green here is acetylated histones. This very dense spot here is this highly condensed pericentric heterochromatin, and so it makes sense. Heterochromatin is very condensed and silenced, And so it makes sense you would see a ton of methylated histones there. And then here, this same section is stained for acetylated histones, and you're not seeing anything. This is all red. The red is staining DNA. So you've got this heterochromatin with no acetylated histones. But you do see green over here, and these different bands are different DNA, different parts, different segments of the chromosome. And these are more associated with gene expression over here. Okay? Make sense? Okay. So there are non-histone proteins that are involved with formation of different types of heterochromatin. So we've talked a lot about histones and modifications of histones and the type of modifications of histones that are associated with heterochromatin, but it's not only about histones, there are a lot of other proteins involved. So two that the book brings up are HP1 and PCG, which polycomb group proteins. And they're both proteins that tend to localize in regions of heterochromatin and different types of heterochromatin. So if you look at the whole genome, you can start to see that some regions of heterochromatin, I don't know, 15% bind to HP1, some bind to PCG, some bind to both. And then there's this whole bunch of like 30 or 40% of heterochromatin that doesn't have HP1 or PCG, but has some other heterochromatin proteins involved. And so this is just to explain to you that there are certain proteins involved in formation of heterochromatin, formation and maintenance of heterochromatin. So HP1 binds to lysine 9, histone H3 with lysine 9 methylated, and it's associated with epigenetic repression and also associates with lamin B receptor. Does anyone know what a lamin, nuclear lamin is? Nuclear lamin is part of the usually like cytoskeletal structure of the nucleus. So it's a good marker for a nucleus, for the nucleus, nuclear envelope. So how do you think it binds to histone tails? How do you think HP1 binds to histone tails? Through what kind of domain? I'm going to pick someone. So my question before I pick someone is, it binds to methylated lysine, and it's associated with epigenetic repression and the lamin B receptor. How do you think it binds to histone tails? And I'm asking Navdeep. There you go. Yes, what do you think? I'm telling you it binds to methylated histone tails. What kind of domain do you think it might have? Right. Right. It actually has, HP1 actually has 2 chromodomains. Great. 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 So this is the kind of questions I might have on an exam where I'm actually asking you to reach back into what I taught you already and integrate it with some other things that I taught you. Okay, and then the polycomb group proteins, they're also associated with long-term maintenance of repression. Okay, so non-histone proteins associated with heterochromatin formation. And you can think about this, if you know something about the different types of proteins and what they're associated with, you can start to use them as markers for different types, like if different types of genes are located in different areas of heterochromatin. Okay? Okay. Polytene chromosomes, they decondense in areas of transcription. So this is showing it in polytene chromosomes, which are these Drosophila chromosomes, but which have a certain structure. And that's where a lot of research has been done, but you can apply it across the board, right? Are you going to have highly condensed DNA in areas of transcription? Probably not, right? Condensed DNA is not conducive to gene expression. So this is just showing these polytene chromosomes where they had, I think they did electron microscopy with some way of detecting RNAs, probably with poly A or I don't know what with. but they're detecting RNA here in each of these black dots. And so notice this banding is gone in this section here. And so this is this chromosome puff they're talking about. So there's an absence of condensed bands of DNA where the RNA is being transcribed. If you could see the DNA, you can't actually see the DNA. What do you think it would look like? Here you can just see there's no dark condensed bands in this region. But if I gave you a pencil and I said, come up here and draw in the DNA, what would it look like? Would it be condensed, or would it be Ashley Miller? Nope. Where's Ashley? There's Ashley. I think she's sick. Okay. Rachel May. She's sick as well. Is there a virus going? I hope everyone's better by Monday, or Tuesday, or Wednesday, or Friday. How about Khan? I saw Khan. There he is.
Is it fragmented?
Fragmented? You think the DNA? So I'm asking what the DNA would look like. Do you think the DNA gets removed from the chromosome? No.
No, I think it just opened up.
Okay, so it opens up. It opens up, and it's going to have this looped out extended conformation in that region. Okay. Okay. Yes.
Actually, can you go back to the previous slide? I just wonder what are those like condensed bands consist of?
The condensed bands. What do they consist of? They're DNA.
All DNAs? Or
It's all this. So this is what I mean. Obviously, there's proteins and stuff, but it's a chromatin. It's the structure of what a chromosome looks like. These polytene chromosomes look like in Drosophila.
So those dots only consist of DNAs but not proteins?
These dots are RNA.
Okay, RNAs. Okay.
Yeah. So it's a marker for gene expression. Okay. Any other questions about this? Yes.
So what are they marking? Like what do you
What they're trying to show, what the reason I put this slide here is because you've got these area of this condensed DNA throughout the chromosomes all over here. But over here is where your transcription's happening, and notice there's none of this condensed DNA here. There's none of these condensed bands. And so what I'm trying to illustrate is that where transcription is happening, you've got decondensed DNA. The DNA is going to be open and looped out. Make sense? Yeah.
Shall we visualize that? Is that an additional loop on top of the Lamp-Rush structure?
This is not Lamp-Rush. The Lamp-Rush was one Lamp-Rush is in the, okay, let me take this back.
Am I confusing concepts here?
Well, no, because there's kind of two models for studying chromosome structure. So the Lamp-Rush model is coming out of the amphibian oocytes, and it's just a very magnified model of what all eukaryotic chromosomes do look like. So there is looping out, but these are polytene chromosomes, which are these Drosophila chromosomes, which are which have a little bit of a different look to them. But the point is, so you can see these regions of very dense DNA, but the point is they're gone in areas of transcription. Make sense? And that's why I put in this, if you could see the DNA, what would it look like? And if I had a marker, I would probably like loop a big thing out over here, a huge loop. Yes?
Is RNA usually circular?
Is RNA circular? No, this is just a marker for RNA. It's not, you're not actually seeing RNA here. So usually with electron microscopy, they use some sort of metal dust that will sit on whatever you're trying to look at, and then you look at the shadowing, and so you're kind of just seeing the marker for the RNA. It's not actual RNA. Yeah. Yes?
How come there's still like RNA in the condensed section?
Does RNA, well, so you're seeing this, right?
Yeah.
Okay, so my assumption in looking at this picture is RNA is not going to stay where,
Okay.
you know what I'm saying? So it's going to diffuse outwards a little. Yeah. So the question was, why are we seeing RNA over here? But I don't think that the RNA is coming from there. I think it's diffused that way.
Okay.
Any other questions about this? Sometimes when I try to illustrate a simple concept, it becomes confusing. Huh? Okay, so let's move on. So each of the 46 interphase chromosomes occupy their own territory in the nucleus. Did you guys know that? Isn't that cool? So this is detected by FISH, fluorescence in situ hybridization, where you have these probes that match certain sequences in DNA, so you can tag different genes or different regions of a chromosome, and they're conjugated to some kind of fluorescence, so you can see them. Yes?
Do each of the chromosomes carry different genes, or all of them carry the same gene?
That is an awesome question. Okay, so each of your chromosomes have their own sequences. So colon 5 is on chromosome 11 and only chromosome 11. I can only tell you that one because I work on it. Every gene has its own location in the genome. Homologous chromosomes will carry the same gene. So both copies of chromosome 11 will both carry colon 5. Yes?
Now, is this model accurate for each nucleus? Like every nucleus will have like the same pattern or position relation with other?
Will every nucleus in an organism have the same pattern or position?
Association with similar or surrounding?
So the question is, what are they? So your real question is, what are they associating with that's keeping them in their certain location? Yes.
Also, we said that transcription happens in the center of the nucleus.
Mm-hmm.
So, one of the chromosomes on the periphery has a gene that needs to be expressed.
Mm-hmm.
Mm-hmm.
Yeah, it does. So what you're seeing here, you're seeing a two-dimensional picture, but the nucleus is not two-dimensional. And so it's almost like if you did a cross-section, you would see different chromosomes in different places in the nucleus. But the center, genes, and as you'll see shortly, genes do move towards the center as they're being expressed. I don't know if I don't know. So the question would really be, what determines where a chromosome is localized? And you would think if it's interaction with, and I think it's not really known. I feel like I read something in the text about how how it's not really known, but I don't know. So we should look that up. I think that would be really interesting to look up. Yes?
So each cell has 46 chromosomes, right?
Each cell has 46 chromosomes.
And each of these chromosomes carries their own gene.
Each chromosome carries a set of genes that make up the full genome when they're all combined together.
But their DNA are all the same.
The DNA of the organism is the same from cell to cell. How many genes do we have in the genome? Who knows these kinds of numbers? 20,000? Let's say we've got 20,000 genes. I forget these numbers. I just like throw them right at my head. Let's say there's 20,000 genes. Those 20,000 genes are distributed across the 23 chromosomes. So you have homologous pairs, but yeah. So they're distributed across. So you might have 1,000 here, 10,000 there.
So the genome is just the total of all the genes found in all the chromosomes.
The genome is made up of all the genes in all of the chromosomes together. Yes. Yes. Yes.
Aren't one of the homologous chromosomes deactivated though? Like if you have two X, one's deactivated, is it just?
Yeah, so you do have X inactivation, so you don't, yeah, but don't worry about that here. You're going to see both, because look, you've got 4, here's one 4, and there's another 4. Here's one. Where's the other one? Maybe I'm wrong. Maybe that's just part of 4. They're right next to each other. I think it's just, yeah, I don't know that it's 2, but I think that the staining is like kind of behind each other. But either way, the whole point is they have their own location in the nucleus. Okay? You've got, you know, it's like you've got your house, I've got my house, they've got their own territory. I thought that was cool. Okay. So gene-rich regions of the chromosomes localized towards the center of the nucleus, and that comes with what you were saying back there. And heterochromatic regions localized to the periphery by associating with a nuclear laminate. And that's what I said before about HP1, how it binds to a nuclear laminin receptor. And so that's how, man. I always have questions that I put in and then I answer them. But you know, that's good because now that I'm picking people at random, you'll be happy that I answered them before. So the point is, so if you look here, the regions, gene-rich regions are green, and see they're all in the towards the center, and the heterochromatin is associated here with the nuclear lamina, which is the red, and then some of the intermediate, not too high percentage gene regions are in yellow. So how does heterochromatin know to localize to the correct region of the nucleus? For those who weren't listening when I just gave you the answer, or I started to give you the answer. I'm going to ask Catherine Buck.
You said that the HP1
Good, very good. So HP1 is one of these proteins involved in heterochromatin formation, and it binds to the nuclear lamina, so that would be how heterochromatic regions tend to localize near the periphery. Okay? Yes?
Are the heterochromatic regions only the centromeres and the telomeres?
No. So centromeres are pretty much always going to be heterochromatin, but there are other regions of the genome which will also, or of a chromosome that will also be heterochromatin. Any other questions? Okay, so the position of the gene changes depending on whether or not it's being actively transcribed. So this is an experiment that was done where they stained an area with a specific gene. I think it wasn't, was it a specific gene or was it? Doesn't matter. They stained an area with a specific gene, and then they stained the rest of the chromosome green. So the red dot is the gene of interest. And when they turn the gene on, look at what happens to the red dot. It migrates to the center of the nucleus. So here it's at the periphery, and here it's at the center. And so this is a picture of what you're seeing. There's the specifically marked gene, and you get this looping out and movement of that gene towards the center of the nucleus. Okay? I think that might be very close to it. So annoying. I'm just being angry at the shadows from the lights. So genes, they move to different regions of the nucleus based on their activity, active transcription versus repression. So repression genes are always going to be localized towards regions near the nucleus, and active genes are going to be towards the middle, right? So here's your nuclear envelope, and here's your active genes. So they loop out, they kind of go to this area of active transcription. And let's say you have a gene that's repressed, it's going to go towards these areas that are more for silencing. Who knew? Questions? That's it. So we're early today. We could use this time to talk, to chat, have fun, whatever you want to do. Let me just stop recording.